Hello everyone and welcome back to the channel. Today we re-journeying back in time to unravel one of the most pivotal moments in scientific history, the discovery of the structure of DNA. It's a story of genius, rivalry, controversy, and a breakthrough that fundamentally changed our understanding of life itself. So grab your lab coat and let's dive into the story of the double helix. Our story begins in the mid-20th century. Scientists knew that something inside our cells was responsible for passing traits from one generation to the next. They called it the hereditary molecule. They even knew it was likely a substance called deoxyribonucleic acid, or DNA. But what did it look like? How did it work? This was the billion-dollar question, and labs across the world were in a frantic race to find the answer. In one corner, we have the Cavendish Laboratory at Cambridge University in England. Here, a brilliant but brash young American biologist named James Watson teamed up with an equally brilliant but more reserved English physicist, Francis Crick. Watson was obsessed with the gene, while Crick was an expert in using X-ray crystallography to figure out the structure of molecules. They were an odd pair, but their combined ambition was unstoppable. They didn't do many traditional experiments themselves. Instead, they were master model builders, piecing together clues from other people's research like detectives solving a complex puzzle. Meanwhile, just a short train ride away at King's College London, another team was hard at work. This team included Maurice Wilkins and a remarkable scientist named Rosalind Franklin. Franklin was an exceptionally skilled X-ray crystallographer. Her expertise in this technique was second to none. She was meticulously and cautiously working to capture images of the DNA molecule using X-ray diffraction. This process involves shooting X-rays at a crystallized form of a substance. The way the X-rays scatter creates a pattern on a photographic plate, and from that pattern, a skilled scientist can deduce the molecule's three-dimensional structure. Franklin, along with her student Raymond Gosling, managed to produce some of the clearest and most detailed X-ray diffraction images of DNA ever seen. One particular image, which has become famously known as Photo 51, was a stunningly clear picture showing a distinct X pattern. To a trained eye, this was a huge clue. An X pattern like that strongly suggested that the molecule had a helical or spiral shape. Franklin noted this in her lab books, but being a thorough and careful scientist, she wanted more evidence before she announced a definitive structure. She was cautious, methodical, and rightly so. The stakes were incredibly high. This is where the story takes a controversial turn. The environment at King's College was not exactly collaborative. There was tension between Franklin and her colleague, Maurice Wilkins. In early 1953, without Franklin's knowledge or permission, Wilkins showed Photo 51 to James Watson. Once seeing that image was a eureka moment. He immediately recognized the helical structure. He sketched the pattern on a newspaper and rushed back to Cambridge to show Crick. The X from Photo 51 was the missing piece of the puzzle they needed. It confirmed their suspicion that DNA was a helix. But there was another crucial piece of information they obtained, this time from a report summarizing Franklin's work. This report, which they also saw without her consent, contained her precise calculations of the DNA molecule's dimensions. It gave them the exact size and symmetry of the DNA unit cell. Armed with Franklin's data and another key clue from a biochemist named Erwin Chargaff, they were ready to solve it. Chargaff had discovered that in any DNA sample, the amount of a base called adenine, or A, always equaled the amount of thymine, or T, and the amount of guanine, G, always equaled the amount of cytosine. See, these are now known as Chargaff's rules. Watson and Crick put it all together. They realized DNA wasn't just one helix, but two strands, twisting around each other like a spiral staircase, a double helix. The rungs of this staircase were made of the base pairs. Using their model building skills, they figured out that A always paired with T, and G always paired with C. This pairing explained Chargaff's rules perfectly, and also suggested a brilliant mechanism for how DNA could copy itself. The two strands could simply unzip, and each strand could then serve as a template for creating a new, complementary strand. It was simple, elegant, and it explained everything. In April 1953, Watson and Crick published their findings in a short, one-page paper in the journal Nature. It began with the now famous, understated line, We wish to suggest a structure for the salt of deoxyribonucleic acid. In the same issue, papers from Wilkins and Franklin's teams were also published, providing the experimental data that supported the model. The world was about to change. The discovery of the double helix opened the floodgates for modern genetics and molecular biology. It gave us the blueprint of life itself. Suddenly, we could understand how genetic information is stored, copied, and passed down. It paved the way for everything from genetic engineering and DNA fingerprinting to gene therapy and the Human Genome Project. Every time you hear about breakthroughs in treating genetic diseases or see a crime solved with DNA evidence, it all traces back to this one foundational discovery. In 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine for their discovery. Tragically, Rosalind Franklin was not included. She had passed away in 1958 from ovarian cancer at the young age of 37. The Nobel Prize is not awarded posthumously. For decades, her critical contribution was largely overlooked and understated. Watson's own book, The Double Helix, painted a rather unflattering portrait of her, which further cemented a narrative that downplayed her role. However, in recent years, history has begun to correct itself. The scientific community and the public now widely recognize Rosalind Franklin's essential and invaluable contribution. Her Photo 51 was not just a small clue. It was the critical piece of evidence that unlocked the secret. Her story serves as a powerful reminder of the challenges women have faced in science, and the importance of recognizing the contributions of every member of a research effort. So the discovery of DNA was more than just a scientific breakthrough. It was a human drama filled with ambition, competition, and a quest for knowledge that defines the very spirit of science. The elegant double helix is not just a molecule. It's a symbol of life's complexity and beauty, a twisting ladder that connects all living things. It's the code that writes the story of who we are, and it all started in a few labs in England, with a group of brilliant, flawed, and determined scientists who changed the world forever. What do you think is the most fascinating part of this story? Let me know down in the comments. Thanks so much for watching. If you enjoyed this journey into scientific history, be sure to hit that like button, subscribe to the channel, and ring the bell so you don't miss our next video. See you next time.